Okay, so today we're going to finish up talking about the somatic nervous system. So when we left off, we had talked about motor units and how motor neurons innervate muscles, skeletal muscle, create depolarizations and action potentials in muscle fibers. We had looked at the steps of that process at the neuromuscular junction, remembering that basically this is working in a very similar way to the synapse, which we have already studied. Just we're thinking about a couple new vocabulary terms and a couple more specifications because we're thinking about a new system. So we're going to start with a little recap in video form, and then we will continue. Let's see if this works when I click on it this way. Yeah, right.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in 2 ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
All right. So that's our overview of what we talked about last time. All right. And the next thing we have here on a table is just a big comparison of the parts of the nervous system, which we've now talked about pretty much in full. So we're looking at all of our efferent nerves. So let's say the motor division of the nervous system. So coming out of the central nervous system, going into the peripheral nervous system. So going out to your body. We had a split there in our efferent division and these motor nerves. We either have autonomic nerves that are helping you do homeostasis, regulation of the body, operating in relative secrecy, or we have our new somatic nerves, which are going to skeletal muscle, so things under voluntary control. In our autonomic nerves, those homeostasis involved nerves, we had a second split between parasympathetic nerves, our rest and digest part of the system, and our sympathetic nerves, our fight or flight part of the system. And in this table, we can see the description of the neurons in the pathway, the description of the effector organs for each of these branches, the neurotransmitters involved between the postganglionic neuron, if we're thinking parasympathetic or sympathetic, or the motor neuron, if we're thinking about somatic, and the effector organ for each of these. You can see that receptor type for each of these that would be on the effector organ, receiving that signal, receiving that neurotransmitter. We can see some of those effects here. So this is a big summary table to pull it all kind of back together. So we actually are going to do one of these checkpoint questions next. So to review, we want us to check in, make sure that we know which neurotransmitter we were talking about here in this most recent part of our nervous system. So what type of neurotransmitter were we thinking about when we're thinking about our somatic nerves, our motor neurons at that neuromuscular junction as we're getting to the muscle? We want to know what type of neurotransmitter what type of receptor that neurotransmitter is binding to. Okay, take another second or two to pick a choice. And we'll take a look. Okay, cool. Yes, the answer is acetylcholine. So our motor neurons are releasing acetylcholine at that neuromuscular junction, just like all of our preganglionic nerves were in our autonomic nervous system, whether sympathetic or parasympathetic, and our parasympathetic postganglionic neurons, all releasing that same neurotransmitter. When we're thinking about the receptor, cholinergic receptors receive acetylcholine. That's where those names come from. Adrenergic receptors receive epinephrine, which is also known as adrenaline. Okay, so adrenergic, adrenaline is for epinephrine and norepinephrine. We had two types of cholinergic receptors. I didn't make you pick between them on this question, but we had muscarinic cholinergic receptors, which are going to be specific to the junction between a parasympathetic postganglionic neuron and its effector organ, which will be smooth muscle, cardiac muscle, or glands. Our nicotinic cholinergic receptors are the one we're going to see everywhere else. So in those ganglions, those synapses between preganglionic and postganglionic neurons, we'd see nicotinic cholinergic receptors. And on our muscle fibers, we're going to see those nicotinic cholinergic receptors again, because they're pretty simple. They work fast. They're ionotropic. They just let an ion in as soon as that neurotransmitter binds. The muscarinic receptors are the ones that are more complicated. and G protein coupled. So at the endpoints of our parasympathetic branch of the autonomic nervous system, we can have kind of more complicated long-term effects because of that different type of receptor. Okay. Cool. So to target in, finish up, wrap up, tie it all together, our neuromuscular junction, and take a look at some toxins that can actually affect how your muscle functions because they are affecting what's going on at this neuromuscular junction. So this first toxin that we're thinking about stimulates release of acetylcholine. So when we say it stimulates acetylcholine, this is a toxin that we see in black widow spiders. We mean that we're going to release a lot more of that neurotransmitter. So we're going to see more neurotransmitter here in this synaptic cleft at the neuromuscular junction. So, oh, there's that name of that toxin, latroxin, is in black widow spiders. So when they bite you, this is what happens. Okay, I want you to take a guess. What do you think is going to happen to a muscle if we have that motor neuron releasing extra acetylcholine? So take a second and think through the steps of what's happening here at the neuromuscular junction and try to predict what's going to happen on that muscle end. I give you a 50-50 shot. Take a look. Okay, yeah, most of you got this right. So the muscle is going to spasm in response to this latroxin. So what we have happening here is when we have more release of acetylcholine, we end up with more acetylcholine here for those receptors to receive. So this basically means that we're going to trigger a stronger response in the muscle cell. Okay, so we're going to see more sodium going in as more of these receptors are getting more acetylcholine, which means that we're going to see more action potentials happening kind of for a longer period of time because they're going to keep getting triggered kind of over and over. And so your muscles is going to seize up basically in response to all this extra acetylcholine that's sticking around. Okay. So this causes muscle spasms and eventually causes rigidity. So you're going to be like so clenched up that you're essentially going to be paralyzed because your muscles are going to be stuck in this contracted state. It's going to be super stiff, rigid, contracted across your body. Our next toxin, protoxin, found in venomous snakes, inhibits acetylcholine release. So this next toxin is doing basically the opposite thing, which is actually also dangerous, right? So we're inhibiting acetylcholine release. So we're stopping acetylcholine from coming out of our motor neuron, even when we have a signal coming through that somatic nervous system, right? So you get an action potential coming down the nerve, you're trying to clench your muscle, you just can't because of this protoxin, okay? So this causes, I added too many actions, sorry. This causes flaccid paralysis. So this is basically a different version of being paralyzed, right? Instead of being all clenched up and stuck rigid because of these muscle spasms, basically just stuck out and loose in all these muscles because you don't have the ability to get that neurotransmitter out there in order to actually contract the muscle. Our final example of a toxin here is curare. So curare is a toxin found in plants. So this is the kind of toxin that goes on things like poison arrows. Its role, it does, is it blocks nicotinic cholinergic receptors. So I'm going to have you predict again what the result there is going to be if you block those receptors. And you got the same two answer choices. Take a look. Okay. Again, yeah. So the muscle is going to relax, just like with the latroxin from that black widow spider. So if we go back and take a look, go back a slide. Can. Okay. So if we take a look just at that diagram one more time, right? We're attacking the same system, but later on in the process, right? Oh, sorry, just like the crotroxin, sorry. So we're attacking this neuromuscular junction on the muscle side now. Curare is actually essentially like a antagonistic competitive inhibitor here. It blocks our nicotinic cholinergic receptors. So even though our acetylcholine part is working just fine, we're releasing the right amount, nothing's going wrong there on the neuron side, we can't get our sodium into the cell anymore on the muscle side. We need that sodium to come into the cell so that we can have the depolarization causing an action potential. So this acts as a muscle relaxant. Now, this is a problem if you have lots and lots of curare, but we've actually found a way to kind of adapt this poison to act as just a muscle relaxant for like therapeutic uses. So actually we do kind of use this in medicine because these things are kind of dosage dependent, right? So if you give someone just a tiny bit, you don't affect all of these receptors. So you can kind of just relax the response of the muscles rather than completely paralyze them. So this is actually kind of useful in the present day. All right, so now we're going to move into looking at the muscles themselves and specifically the skeletal muscle. So we're going to go into our first packet of muscle slides. We're going to start out by just looking at the general anatomy, the general shape of what a skeletal muscle is going to look like from a kind of microscopic level, not like what you would see in lab. Okay. So when we're thinking about any named muscle, we're going to have layers to it, right? So we talked about the fact that we have motor fibers innervated by motor neurons, as we were thinking about the somatic nervous system that innervates skeletal muscle. But if we're looking on a larger level, we're going to see some layers before we even get down to that muscle fiber level. So the muscle fibers are really cells, long skinny cells, rather than the muscles themselves. So we have some connective tissue surrounding our muscles. So every time you see like this "my" in a word, you should suspect that this has something to do with a muscle. So our epimysium, epi means like around the edges, kind of, is going to be a layer of connective tissue that thinly surrounds the outside of the muscle. So we can see that this is surrounding many, many muscle fibers and also many little bundles of muscle fibers. Okay, so it's going all the way around. Diving deeper, we're going to have the perimysium. So this extends into the muscle body. And this is going to separate out little bundles of muscle fibers called fascicles. So you can see that each of these muscle fascicles is actually made up of several different little muscle fibers. So we're grouping them together here with the perimysium. Okay? Each fascicle, unlike in this image, actually contains hundreds to thousands of muscle fibers. Okay. Diving even more deeply, within each of those fascicles, we're going to see connective tissue surrounding between each of those muscle fibers, which are going to be our individual cells. So this is the endomysium. So think endo like inside. So it's inside the muscle, inside the fascicle. So it's holding together these muscle fibers, essentially. Now, some other words we want to know when we're thinking about our muscle cells. Okay. So we heard the word sarcolemma in that little video at the beginning. So the sarcolemma is the plasma membrane around a muscle fiber, so a cell specifically. So we've like renamed the membrane of this cell. So our sarcolemma is our plasma membrane, right? So that's just the membrane of the cell. Okay. Our muscle fibers, our muscle cells, are special in that they actually have multiple nuclei. So you can think of them as almost like many cells all linked together into one big long cell. Okay, so we have these many nuclei down the length of our muscle fiber, which is a single cell. Okay. Then we have one more new word here, which is our sarcoplasm. So we've also renamed the cytoplasm. So that is really just the same thing, but now we're being specific that we are in a muscle cell. So sarco should also indicate to you that we're thinking about muscle cells specifically here. So mysium, we had it for sort of bigger layers of muscles. Now the sarco, we're down at the cell level. I'm looking at that muscle fiber. So again, we're at the cell level. So we can see renamed sarcolemma is our membrane of the cell. Our sarcoplasm was our renamed cytoplasm on the inside. Okay. We are going to have some more special adaptations inside that cell that don't look like our normal organelles that we look at in our basic animal cell. Okay, so we're basically also going to have a specialized structure analogous to our endoplasmic reticulum that you would have seen in a basic cell in one of your other classes. Now we're going to call it the sarcoplasmic reticulum, right? So we've just, again, renamed it, specified that it's in a muscle cell. Yeah. And its structure is such that it's kind of along the inside of that sarcolemma. So it's along the inside of that renamed plasma membrane, but it has a bunch of extra kind of swellings called lateral sacs, right? So we've got these like swellings here, our lateral sacs in this sarcoplasmic reticulum that are storing calcium. So calcium is going to be important for muscle function, just like it was important for releasing neurotransmitter in our neurons. Okay, so it's going to have a new special job here in our muscle cells. Next to these lateral sacs, we're going to have a structure that I don't have an analogy for in a normal cell. This is our T-tubule. So our T-tubule is a T down into the cell. So T is for transverse tubule. So it is a tube diving into the cell from that sarcolemma, from that plasma membrane. So you can see it going down in and neighboring those lateral sacs filled with calcium from the sarcoplasmic reticulum. The overall structure that this is going to create inside the cell is that we're going to have this tube diving down into between the, let's say into that sarcolemma. So across that plasma membrane. And on either side, we've got sarcoplasmic reticulum filled with calcium. Okay. And this structure as a whole is going to be something called a triad. So you can see this kind of T shape to it. We've got a T with two storage areas for calcium on either side. This is going to be important because as we see muscle cells getting triggered to contract so that you can actually move stuff, this is going to play a role in how we're transmitting that signal and telling the muscle fiber to contract. So we're going to see the action potential traveling down these structures, releasing calcium, telling your muscle that it's time to get shorter and pull on stuff. We'll talk about the steps of that in a little bit. Okay. Can also refer to the muscle fibers, essentially. Within them, we're going to have these additional fibers called myofibrils, which are made up of two proteins. We're going to have thick filaments made of a protein called myosin. It is physically thicker. That's why it's a thick filament. We're going to have thin filaments made of a protein called actin. They are literally thinner, hence their name. They're going to form structures called sarcomeres. So a sarcomere, it's going to be like these blocks that we see along our muscle fiber, along these myofibrils. So we're going to chunk out our muscle fiber, not into different cells, but into sarcomeres, into chunks. So these are what are going to ultimately shorten when our muscle itself contracts. These units are each going to get shorter. And I put up an activity yesterday afternoon that you have until end of day Friday at 5 p.m. to do, is going to have you pinpoint some of the features of the sarcomere and just like identify them. We're going to run through them today, but that's where you can practice that and think about that a little more deeply and kind of more slowly. It's not timed. You've got unlimited attempts, but I want to make sure that you know the structure of the sarcomere. So as we talk about why it's getting shorter, what parts are getting shorter, you really understand that. Okay. Because we got a long list of pieces to the sarcomere, right? So I'm going to talk through them, but you want to spend some time looking at them on your own as well. Okay. So our sarcomere is our unit of the myofibril, right? So we have them inside our muscle cells. I'm going to circle out a sarcomere here. I'm actually going to talk about these in a different order than listed here because I think they make a bit more sense in a different order. At least I'm going to just walk you through the order I think about them in. Okay. So here we have a sarcomere. So I'm going to actually start at the end because our sarcomere is measured from Z line to Z line. Okay. So this is our sarcomere. This is that unit I was talking about. Okay. So those are the endpoints of our sarcomere. And what those endpoints are doing, those Z lines, is they're stitching together all of our thin filaments. So our actin was the thin filament, right? That's the protein that makes it up. So this is the actin, the thin filament. And the Z line is securing all those little thin filaments to each other at both ends of the sarcomere. Okay. So those are our boundaries. Okay. The next thing I like to think about is the M line, because now we've thought about thin filaments, we can think about thick filaments. Okay. I like to do the lines first and then the bands, basically. Okay. So actin is our thin filament. Myosin is our thick filament. So our thick filament is this one that looks kind of like wheat. That's the myosin. Okay. And conveniently, myosin, the thick filament, is bound together by the M line. So M for myosin. So that's going to be like the center point of our sarcomere. So this paler red line down the center is our M line. So we go out to the Z line to the left, out to the Z line to the right, makes up our full sarcomere. Okay. Now that we've talked about the endpoints, those Z lines and the midpoint, the M line, we can talk about the zones and the bands. Okay. So the zones and the bands are going to be referring to basically colored patches almost that you would see on a slide. If you put this under a microscope, is how they started to be named, which is kind of why their names can seem a little bit random. But they're important because they do correspond to specific parts of the proteins, and specifically, they're going to tell us zones where we have our filaments overlapping or not. And if not, we're going to be able to identify which filament is present. Because of that, we're going to see that some of these zones are going to stay the same length, no matter how long or short that sarcomere gets. And others are going to change size, depending on whether we're in the contracted state, so the short state, or a relaxed state, that longer state. So when we're looking at these, we can do these in order now. All right, let's focus on that myosin for a little longer. So that myosin, we saw was linked together by the M line. It's our thick filament. It's this one that looks kind of like wheat, okay? Anywhere you have myosin, it's your A band. So you can see the A band here is from the end of the myosin to the other end of the myosin, okay? So anywhere you see myosin, you'll have this sort of dark band on a slide, okay? So the A band is where you see myosin. And this means, actually, because neither of our proteins actually change size when we see our sarcomere contract, when we see the sarcomere shrink, because our myosin doesn't change size, we're going to see that that A band actually is not going to change its total length when we see our muscle contract. Okay. The H zone, however, will change size. So the H zone is referring to an area where we have myosin, but it's referring to the area where we have only myosin. So the H zone is where we have only this thick filament and no overlap. So when I say overlap, right, when I pull out this H zone in the center, right, you see only that one little orange line, versus if I were to go to the side, right, here on the side, in the rest of that A band, you have both that wheat-like myosin and these blue-purple extensions of the actin, right? So that would be part of the A band over here, but not part of the H zone, because the H zone has only myosin and nothing else. Okay. So that's what the H zone is. Now the I zone is our other area where we have no overlap. So the I zone, sorry, the I band is this area over here, right next to the Z line, where we have only actin, so only our thin filament, okay? So as soon as we have overlap, we're into the A band. If we have only actin left at the end, we have a lighter band on a slide, right? And this represents only thin filament with no overlapping. So I think it's called the I band because you imagine taking a Z line and just that non-overlapping I band on either side, right? You kind of see an I here on this side. So I suspect that's why they named it this way. Okay. Now we're going to talk about the structure of each of those filaments. So our thin filament is actin. Actin is one of our contractile proteins. So actin is going to be involved in contracting our muscle. So actually shortening it, making it smaller. Okay. So because it's a protein, it's made of many parts. Okay, so our little individual molecules, let's say, are made up of these beads called G-actin. So like globs of actin, G-actin. They look kind of like olives. They're each going to have a little site for binding on to our other contractile protein. So we have a site where actin is going to bind to myosin. So notice we're looking at actin right now, but we have a myosin binding site. So you're going to want to be careful about that. It's going to be named for what it's binding to, not named for what protein it's on. So on actin, we're going to have a myosin binding site for myosin to bind to. G-actin gets linked together into F-actin, longer, right? We've made a string. And then when we're looking at a long strand of actin, we're looking at a double helix kind of structure swirled around. Now, on top of the actin, we have some proteins that are going to be really important for the behavior of actin and how and when it's allowed to bind to myosin. Okay. So first, we're going to think about tropomyosin. Okay. So tropomyosin is in this bottom image. It's this long spaghetti structure. If you look carefully at the image, you're going to note that in this picture, that tropomyosin is covering over each of these myosin binding sites, right? So it's going across that pit on each of these little olives, right? So when tropomyosin is lying over that binding site, it's basically blocking it off, right? So if we have tropomyosin covering the binding site, like we do in that image, there's nowhere for myosin to bind to. So we're not going to have our two contractile proteins binding onto each other, holding onto each other, and allowing the muscle to contract. So tropomyosin is important because otherwise we basically end up with our proteins kind of stuck together all the time. Okay. So tropomyosin covers these binding sites in this first state of our actin. Okay. Troponin is another protein that's going to be important for regulating how our muscles contract, when they contract, when our actin and myosin can bind to each other. So troponin is this little snowman thing sitting on our actin, okay? And the troponin is actually touching that tropomyosin, right? So this is a real connection here, okay? It's kind of connected there. And specifically, one of the bits of the snowman, the T part, is attaching to tropomyosin, okay? And this means that troponin, as it changes shape, if it changes shape, when it changes shape, is able to push and pull on the tropomyosin and either pull it off those binding sites or let it go back and cover over those binding sites again. So it also has a point where it's attached to the actin, right? So it's got a base where it's attached to our protein, right? So like the groundwork. It's got a part where it's attached to the tropomyosin. And the other part it's going to have is a part that binds to calcium. Okay, so calcium is going to be important for muscle contraction. And where it's actually going to functionally help with that muscle contraction is when it binds to our troponin complex, because that's going to initiate a shape change that moves the tropomyosin, it's touching the troponin, and allows actin and myosin to bind to each other because this myosin binding site on the actin is now uncovered. Now we're going to be looking at our other protein involved in muscle contraction, our thick filament. So our thick filament is myosin. Okay. An individual piece of myosin looks kind of like a golf club. All right, so it's got a long tail. It's got heads on either end. Okay. On each head, we have an actin binding site. Right, so named for what it's binding to, not what it's on. So on myosin, we have an actin binding site. And we're going to see myosin also has a site for the binding of ATP, essentially. So ATPase is an enzyme that breaks down ATP. So this is ultimately where your muscle is going to be using energy for contraction. It's going to be using it in conjunction with the action of myosin. Okay. Looking at the larger structure, we don't see just this when we're looking at a sarcomere, when we're looking at the thick filament. What we actually see is lots and lots of myosin kind of all together with many of their heads. So many of these golf club projections sticking out. So that's what we're looking at when we see this thick filament. They're not all in a line. They're not all exactly at the end. They're kind of offset from each other. So we've got lots and lots of them grouped together. Overall, the heads, these parts, are going to be pointing towards the I band. So the I band was where we had only actin, right? It was on either side of the Z line, okay? So it's facing towards the actin because these heads are going to be important in binding to the actin as our muscle contracts. And we'll be looking at the details of that in a second. So on that myosin head, on each of them, we have an actin binding site. So it's going to be able to grab the actin, and it's going to have a site where we can bind ATP and use energy. We also refer to these heads as cross bridges, okay? So you can think of them as they're going to cross the bridge to the actin, right? So we're going to form a bridge between our thick filaments, our myosin molecules, and our thin filaments, our actin molecules. We're going to be making that bridge so that we can pull the actin in towards the center of the sarcomere to contract our muscle fiber. Now, you do have one other protein involved here. This is not a contractile protein. It's not going to be involved in physically shortening the muscle. It's basically just supporting everything in place. So you can think of it as a spring. So it's this cable, this spring at the end right here. Okay, so we ignore it when we're naming our Z line, M line, A band, etc. Right? It's just kind of anchoring that myosin, that thick filament to the Z line a little bit in place so that everything stays arranged as it should. So this is just for like structural integrity, structural support, and it's long and stretchy, just kind of springs back into place. So we're not going to think very much about tighten, is basically what I'm telling you, but it is there. So we're going to take a peek, start out by thinking about how muscles actually generate force. So like, how are we contracting? How are we moving stuff? So when our muscles contract, they're going to do so by shortening their sarcomeres. When our sarcomeres shorten, we don't actually have either the actin or the myosin, our two contractile proteins, shrinking. They're just sliding past each other. So we refer to this as the sliding filament model, because you imagine, right, my fingers here on this side, and then second on either side are going to be the thin filaments, the actin. So here's my Z line. My pencil here is going to be the thick filament. So what we have when our sarcomere shortens, I'm about to put this pencil in my mouth, it's going like this. A long straight, and a short straight. Just like that. That's it. That's all that's happening. So we're just having these fibers slip past each other. They're just sliding past each other. And as they slide past each other, those Z lines are coming closer together. So overall, the sarcomeres are shortening, even though neither of the proteins got shorter, which means overall our muscle fiber gets shorter and you can pull on either end. Okay, so that's part of the how. The other part of the how is the cross-bridge cycle. So the cross-bridge cycle is the full process by which that myosin and actin bind to each other, use ATP to create that sliding. So we're going to see that we use energy for that myosin to essentially grab onto actin, pull it towards the center of the sarcomere to make the sarcomere in total shorter. Later on, we'll talk about how we turn those contractions on and off, and we'll talk about how exactly they get the ATP in order to drive that cross-bridge cycle, because different muscle fibers are going to do this in a different way. So sliding filament model, I just modeled to you with my hands, and in that sarcomere activity, I've also linked you to a copy of a Google slide where I just gave you two sets of thin filaments and a set of thick filaments, so you can practice just kind of seeing how they can slide past each other and make a shorter sarcomere without actually shrinking the size of either of those proteins. All right, sliding filament model, we just have these muscles contract because sarcomeres shorten as our region of overlap essentially becomes greater, right? So when I did this, not very much overlap, lots more overlap as I got shorter. Right? So we're going from not very much overlap at the longer state. Overlap increases greatly when we make that sarcomere nice and short. So that's what's happening as these filaments slide past each other. So in the activity, I'm going to ask you to take a look at sarcomeres and practice for yourselves identifying how these zones shorten or don't shorten depending on the size of that sarcomere. All of this is happening due to the cross-bridge cycle. So all the answers to that activity are in your slides, but I do want to make sure that you really try to internalize these, right? Because otherwise you're going to have to just memorize random facts about this, and it's going to make it harder to understand what we're talking about next, which is going to be the cross-bridge cycle. So if you want to be really ahead of the game for Friday, try to find this figure in your book and start to make sense of it, because we're going to be stepping through it. And it could be a lot if it's the first time you see it in class.